Hello everyone. Welcome to this edition of Monday's Ocean Talks. My name is Guilherme. I'm a research assistant at AIMM, and today we have the pleasure of interviewing Dr. Robin Baird, which is a biologist currently with the Cascadia Research Collective in the United States. Dr. Robin has had a very nice and influential career in marine biology. He first got his degree in the University of Victoria in '87, later a PhD in biology by Fraser University in '94, regarding the foraging behavior and ecology of transient killer whales, and later a postdoctoral fellow at Dalhousie University in Halifax, Canada. Throughout his career, Dr. Robin has been able to work in several other projects and initiatives. He has a very prestigious career. He's the co-founder of AIMM, which is an electronic mail discussion list focusing on marine mammal research and conservation. He has authored or co-authored more than 130 peer-reviewed papers, publications in 2 books, as well as several book chapters. So welcome, Robin. It's nice to have you here. And thank you for participating in this interview.
Yeah, my pleasure. Thank you.
So to start, every story has a start. I would like to know, first, we would like to know what got you into marine biology. How did it start? Was there a specific moment in your life where you decided that this was the career you were going to follow?
Yeah, well, when I was a little kid, I always wanted to work with animals. I originally thought that I probably wanted to work with animals in a zoo or an aquarium. I grew up in Victoria, British Columbia, right along the ocean, and was able to go to tide pools regularly. I remember when I was a small boy finding a person in, it was really, really low tide, and there was a person out in one of the tide pools looking around, and several of us went out and were talking to her, and she said she was from the university and she was studying octopus, and you know, I think it just evolved from there. When I was an undergraduate at university, I was interested in birds. I was interested in reptiles and amphibians, and getting specifically involved with marine biology, with whales and dolphins, was something that just sort of evolved during my undergraduate career.
Okay, so you were in the beginning, you were more interested in birds. I also share a bit that passion, and I have also in other interviews you've mentioned that birding is great because it allows you to develop this attention for detail and also for identification. And I mean, if you like animals and if you're interested, then it's very easy to then get, you know, this kind of spreads like wildfire our interests towards any other animals. Then you said that you're interested in cetaceans eventually developed through your studies, but why did it develop in cetaceans, specifically in cetaceans?
Yeah, when I was an undergraduate student at the University of Victoria, we had a ecology class, and as part of that ecology class, our professor took us on a field trip to San Juan Island in Washington State, famous for killer whales and the Whale Museum, and we had a lecture at the Whale Museum by Rich Osborne, and went out to the west side of San Juan Island and actually saw southern resident killer whales swimming by. So he sort of kindled that interest and gave the students an opportunity to get involved in a small nonprofit group that he was involved in starting up in Victoria. So I started volunteering with that group. Eventually that led to an opportunity to get out with Tom Learholm, a researcher from who at the time was working with killer whales in Iceland, and did a lot of work with with sperm whales in Norway. But Tom was going out to look for killer whales and to get some experience working with killer whales, and we went along to help him out. First day we went out to a local lighthouse. We actually had a group of killer whales come by, and so I drove the boat while he was taking photos. that led to volunteering with Mike Bigg at the Pacific Biological Station in Nanaimo, Canada. And we were collecting photos for Mike's long-term killer whale photo identification research. And Mike was one of my earliest mentors. He basically, provided film for us. He gave us a lot of feedback on the photos we were taking. When we witnessed some cases of mammal-eating killer whales feeding on harbor seals, he really encouraged continuing to study that. And so that evolved, that early experience led to a job as a summer research assistant with researcher David Duffus at the University of Victoria. He was working on his PhD at the time. And so I got to spend the summer of 1986 up in Johnson Strait, British Columbia, which is famous for northern resident killer whales. And a combination of those field experiences and mentorship from Dave and from Mike Bigg really helped me develop my interest, use that to start collecting information specifically on killer whale predation on harbor seals, and that eventually led to graduate school. So I approached Larry Dill at Simon Fraser University and basically said I had been out in the field collecting this information and was interested in applying for a graduate degree, and he accepted me for that.
Okay, very interesting and probably very also very familiar to most of us in this field who you eventually start by getting these opportunities in the field as volunteers. And I imagine that it was a very special moment when you were able to drive the boat, not only to participate, but also to learn how to maneuver and position, because, well, it's quite challenging. And then, as you said, this led you also to focus on this topic of the killer whales in this area for your PhD, where you studied the foraging behavior and the ecology of transient killer whales. Could you tell us a bit more about that, about the study itself, also the results, any other interesting facts?
Yeah, well, so this involved going out in a small rigid hull inflatable 4.7 meter boat, choosing myself and one other person, and a lot of it was just trying to be as unobtrusive as possible, trying to follow alongside the animals and avoid disturbing them, in order to watch their natural foraging behavior. And groups of mammal-eating killer whales in that part of the world are relatively small, 2, 3, 4, 5 individuals. Spend most of their time when they're in these small groups hunting. Where I was working, they're feeding almost entirely on harbor seals, although they would occasionally catch harbor porpoises or chase Dall's porpoise, or elephant seals were also on the diet. And that work focused on a few different things, but probably the most important contribution was determining that these small groups of 2, 3, and 4 individuals were basically the optimum foraging group size. So I was able to quantify food intake rates by group size, the per capita food intake rates, and showed that trios, 3 individuals together, had the highest per capita food intake. And that then sort of leads to all sorts of things in terms of the selective pressure on small groups that have implications for why the fish-eating killer whales and the mammal-eating killer whales are socially isolated. Also documented foraging strategies that showed that the mammal-eating killer whales had 2 completely different strategies. One was to go from seal hollout to seal hollout and forage around haulouts. The other was to forage out in open water and generally avoid the haulouts, called these nearshore foragers and offshore foragers, and able to show that these 2 different strategies were effectively equally profitable. And so it's sort of alternative learning the foraging techniques, and those foraging techniques are probably carried on through these lineages. And that was at a time when mammal-eating killer whales were relatively uncommon on the coastline here, and actually over the last 30 years, the population has just continued to increase and increase and increase. So now group sizes are much larger. They're the dominant type of killer whale that people are seeing now in places like Juan de Fuca Strait and around the San Juan Islands, rather than the fish-eating resident killer whales.
Indeed, and yes, because these, of course, there's the situation with the southern residents, which feed mostly on the salmon, and while the salmon is not thriving, the prey of transients is thriving. So, touching a little bit on the southern resident killer whale, situation. Do you have any insights on the situation of the southern resident killer whales? Of course, this is quite a complicated situation with lots of different factors, namely the pollution, but also the whale watching, which is a polemic right now, and also the dams, and just it's a very complex situation. But do you have any insights on the southern resident killer whales? Maybe a personal opinion about specific factors like the whale watching pressure, which is being attributed now as also one of the growing factors in their basically in the impact that they're suffering.
Yeah. Well, I think the southern resident killer whales, like a lot of populations of whales and dolphins, are facing a lot of different threats, as you mentioned: high levels of persistent organic pollutants, vessel noise from large and small vessels, and in their case, the biggest threat, I think, by far is a reduction in their prey availability. And they're salmon specialists, and in particular, they're Chinook salmon specialists. They do eat chum salmon in the fall and coho salmon and steelhead in the winter. But when Chinook are around, that's their preferred prey, regardless of other species of salmon around. So there could be literally millions of sockeye in the area, and they will go after the limited number of Chinook instead. Salmon populations along the west coast of North America have declined dramatically over the last 100 years, and Chinook salmon in particular. And I think that most of the studies have shown that things like survival rates and birth rates are really driven by the availability of Chinook salmon. So I think that other factors are potentially contributing to their sad state, but really, without doing something in the short term and in the long run to restore Chinook populations, I think those other factors like whale watching are relatively unimportant. I'm all for regulating boating around whales. I think it's important, particularly in a situation where there's no limits necessarily on the number of boats. Personally, I'm a fan of limited entry licensing systems for whale watching, like they have in New Zealand, for example. And I think in the long run, those types of systems are the best approach, putting caps on the total number of boats, as well as conditions on approach limits and so on. It's interesting. Whale watching gets a bad rap in this part of the world, but private recreational boaters are much more of an issue than whale watching boats. Over the years, there's been a lot of consistency within the whale watching community, a lot of experience built up by the operators and by the naturalists, and I think that when you compare that to the private recreational boaters who may know nothing about killer whales, may know nothing about the rules and regulations, the majority of the nearby vessel noise and nearby vessel activity is actually caused by private recreational boats. So I think it's worthwhile controlling and regulating vessel activity, whether it's commercial or private recreational, but I think it's important that attention isn't taken away from what the most important problems are, which are the limited Chinook salmon populations.
Yeah, and of course, there's a bit of two sides of this question, because you have the scientific evidence, which is there clearly for impact on cetaceans caused by vessels. But it's also, I feel like a bit of a scapegoat socially. The people are starting to use it more and more. And I completely agree that, of course, if they cannot feed, then other factors are not as important. And we will actually get back to that, to the whale watching topic later. We also here in the Algarve, we conduct boat-based surveys of these whale watching companies. But in Portugal, it's kind of a mixed system. We don't have limited number of licenses everywhere. We have them in the Azores Islands, but here in mainland Portugal, in specific areas, we don't. So moving away a little bit from Washington now, and a few thousand miles to the west onto Hawaii, where most of your research has been focused since 1999, if I'm correct. Could you tell us more about this, this part of your career and how this opportunity to work with the Hawaiian cetaceans presented itself?
Yeah, well, I've been doing a postdoc in Hal Whitehead's lab at Dalhousie in Nova Scotia in the late 1990s. And winters in Nova Scotia are a little bit cold and long. So while I was there, my funding was running out, and I had the opportunity to take a research position with a nonprofit group in Hawaii. So seemed like a good idea at the time. And I moved to Hawaii in December of '98, and when I got there, you know, there's obviously a long history of people studying whales and dolphins in Hawaiian waters. I was based on Maui, which is people have been studying humpbacks off Maui since the mid-1970s. So when I moved there, I took the research program in a different direction, which is instead of overlapping with or competing with the folks that were studying humpbacks or people who were studying dolphins, were studying spinner dolphins. I really concentrated on species that had received little or no research attention. So we started a photo ID catalog with bottlenose dolphins and false killer whales. We started doing some work with pantropical spotted dolphins, slowly started encounters with short-fin pilot whales. So it expanded the research to work on other species. And although when I started it was, you know, I expected to be in Hawaii for maybe a year or two, and I ended up moving away from Hawaii after a year and a half, but continued to return. The project's basically turned into this now 22 year long study. And the work originally started on Maui, but then in 2001 did some field work off of Oahu and off of Hawaii Island, and expanded both the geographic scope of the project among the islands and also the diversity of species that we are studying. So those bottlenose dolphin and false killer whale photo ID catalogs were the basis for a lot of things, but as we started to encounter other species like dwarf sperm whales and Blainville's beaked whales, we started photo ID catalogs with those species as well. Had the opportunity to collaborate with Dan McSweeney on Hawaii Island. Dan had been taking photos, really the only researcher in Hawaii who had been taking photos of species other than humpbacks and spinner dolphins. And from the mid-1980s, he had been getting photos of false killer whales, Cuvier's and Blainville's beaked whales, pygmy killer whales, pilot whales off of Hawaii Island. And Dan basically let us scan all of his slides and negatives and incorporate all of those photos into our catalog. So even though, say in 2003, we only had 2 years of photos of species like Blainville's beaked whales, all of a sudden we were able to add photos going back to 1985, 1986 of those species. And
amazing.
And so inherited effectively some long-term catalogs very quickly. And the work in Hawaii is it's a multi-species effort. We work more or less with every species we encounter, and how much time we spend with any particular group is is really in inverse proportion to how often we encounter them. So for the rare species, the opportunities don't come up very often. So we take advantage of them when they do, and species like pygmy killer whales or dwarf sperm whales, we try to stay with the group until we've got good photos of everyone, and collect other information before we move on. Where species like spotted dolphins and pilot whales, which are the two most frequently encountered species, while we do a lot of work with those, we don't necessarily prioritize them.
Okay, great. That's really interesting, and I think it's of course a smart approach, because Hawaii is an area that gives you relatively easy access to these species that are, well, hardly known, or even at the time, or probably even harder. It's also quite shocking, but more in a funny way, to hear about negatives, because nowadays we live in such a easy digital world. To just hearing the expression "scanning negatives" for me is quite a, I don't know, it's just we're just privileged nowadays. That's very interesting. And you mentioned, of course, that you try to spend more time with the species that are most rare or the least studied. One of them, probably one of the main species you're looking for, is the false killer whale. Why this species in particular? Of course, they are rare, but it seems to be one of the central species in your studies. And why did you feel the need to shine the spotlight more on the false killer whale, or actually look more into them rather than, for example, beaked whales or the kogia, the dwarf and pygmy?
Yeah, well, false killer whales are species I've been interested in for a long time. When I was in British Columbia, before starting graduate school, several of us started up a stranded whale and dolphin program. And at that time, 1987, there was no stranding network in British Columbia. And so I spoke with Mike Baird and asked him about setting up a stranding network. And he said, yeah, go ahead. No one else is doing anything. And one of the first strandings we had in 1987 was actually a false killer whale. And it was the first record of that species for British Columbia. So being able to, when we had that stranded animal in 1987, we documented the stomach contents, we sent tissue samples off for pesticides and heavy metals. And just in writing up a note on that stranding as the first record for Canada, basically discovered how little was known about the species. And this sort of goes back to our earlier talking about birding. Birders tend to be interested in rare species rather than the common ones, although personally as a birder, I'm interested in all of them. But the opportunity to study rarely seen and poorly known species is just being something that has always interested me. And false killer whales in the late 1980s, after that stranding, we had sightings of false killer whales in British Columbia, a lone individual that stayed around for a number of years, and so the opportunity, when I went to Hawaii originally, I knew that it potentially was a good place to see false killer whales. I had been in discussion in the late 1980s with Steve Leatherwood, who was planning on doing some work in Hawaii on false killer whales. And he and Randy Reeves did aerial survey for them in 1988. And so when I got to Hawaii, the area off of Maui, we would see false killer whales come through semi-regularly. And similar to killer whales, they're large social odontocetes. They're cooperative hunters. But unlike killer whales, they're just very, very poorly studied at the time. So we basically, whenever we had the opportunity, would work with them. We originally started doing some suction cup attached time depth recorder work to look at diving behavior. We were collecting biopsy samples for some collaborative work with researchers at the Southwest Fisheries Science Center in La Jolla, California. And the work evolved over time. Our photo ID catalog revealed resightings between years and within years, suggesting a relatively small population. The earliest genetic results that Susan Chivers at Southwest Fisheries Science Center undertook basically were showing that the false killer whales in Hawaii were different genetically than false killer whales elsewhere. And then we also started seeing evidence of fishery interactions, particular individuals with injuries on the leading edge of the dorsal fin, linear injuries on the leading edge of the dorsal fin, that seem to come from getting entangled in fishing gear. And so, small population, genetically isolated or genetically differentiated, and evidence of fisheries interactions, those things all started to come together. We also, the biopsy samples that we were collecting, as well as sending them for genetic analysis, we were subsampling the blubber, sending that to a researcher at the Northwest Fisheries Science Center in Seattle, and found high levels of persistent organic pollutants in those blubber samples. So, my original interest in the species is more the opportunity to study a large social hunter, similar to my work with killer whales, but it became clear pretty early on that the population was under a variety of threats, and if you go forward in time, that population was listed as endangered in the United States in 2012, and we now know and have known for a while that really of all the species of whales and dolphins in Hawaiian waters, it's the most threatened, it's the most at risk.
Right, and of course it came out of a personal interest, but then what the factors you mentioned are enough ingredients for conservation, for local conservation. And also your work has shown that, and also your colleagues, of course, has shown that there are many populations occurring in the Hawaiian Islands, some of which are more associated with the islands, and some of which are with the main islands, and some others are associated with the northeastern islands. This data mostly was obtained through the usage of tags. So namely tags that provide you GPS location of the individuals that were tagged. Can you talk more about the tags, how they work, what kind of data they provide, how they're deployed, and also, if you can, about the reaction that the animals have to the deployment?
Well, one thing I should mention first is that, and it's sort of come up, but most of the work that we do in Hawaii is collaborative research efforts. We have a lot of folks who work with either with NOAA, National Oceanographic and Atmospheric Administration, NOAA Fisheries, other universities, other nonprofit groups, and really what we're able to say about many of these species now comes from this large collaborative effort. The tagging work originally started using suction cup attached tags. Let's see if I have one of them around.
Nice.
And these suction cup attached tags, started using these actually with killer whales back in 1992. But the suction cup attached tags, the VHF radio transmitter and a little time depth recorder, and record information on what the animal's doing underwater. And you can also use the VHF signal to track the animal over time. But they stay on relatively short periods of time. Started using them in Hawaii on pilot whales and blainbills and Cuvier's beaked whales in 2002. False killer whales we tagged, some originally in 1999, spotted dolphins in 1999, but they give very short-term information. You have to recover the tags to get any of the data back. So we started a collaboration with Russ Andrews, who at the time was with the Alaska Sea Life Center, and Russ had worked with Wildlife Computers, a company in the Seattle area, to take their satellite tags and basically configure them for remotely deploying on smaller animals, things other than humpback whales and blue whales, and remotely so that you don't have to capture them. So these satellite tags, this is one of the current or recent configurations are called limpet tags. They basically are deployed with an air rifle, or you could deploy them with a crossbow. They attached with two surgical grade titanium darts. I have one here that basically screw into the bottom side of the tag and attach to the dorsal fin of the animal. And the tags, you don't get them back when they fall off. They sink. They have no flotation material, and that's important to keeping the size as small as possible. Unlike the suction cup tag that you can put on the end of a pole, and you know, if the animal gets close enough, tag it when it's next to the boat. These are deployed when you're at distances of 3, 4, 5, 6, 7 meters away from an animal. And the individuals have to be cooperative. They have to be moving slowly. They have to be parallel to the boat. But the advantage is that once you get the tag on, then it doesn't matter where the individual goes, you will continue to get information back from it. And we, you know, we don't get really long durations depending on the species, but the average duration for one of these tags on a false killer whale is about 45 days. The longest we've had is on a false killer whale was 199 days. And, you know, even in a couple days, they can move 100 kilometers or more. So instead of being restricted to studying them in these limited areas, right? You know, in Hawaii, we work mainly on the west sides of the islands because that's in the lee of the islands from the trade winds. And our study area off Hawaii Island may be 100 kilometers by 30 or 40 kilometers, but the animals may be moving over scales of hundreds of kilometers, either onto the windward sides of the islands or offshore. So the tag work as well, tagging has been an integral part of what we've learned about a lot of species, but it's really being tied into the photo identification work and the genetic work as well, because for any particular group of animals, we may have some tag data from that group, but we also know who the individuals are. We know something about their sighting history, and we know something about their associations with other groups. And in the case of false killer whales, we also have genetic samples from some of those individuals or certainly those groups. And when you tie all three of those together, it revealed the pattern that we now know that in Hawaiian waters, there's 3 separate populations. As you mentioned, a main Hawaiian island insular population, a northwestern Hawaiian island insular population, and then a pelagic population. We know from the tag data that those populations are overlapping in their range, but we know from the genetic and photo ID data that they're socially isolated and genetically differentiated. So the northwestern Hawaiian Island and main Hawaiian Island populations share one of the same mitochondria haplotype, so we know they're closely related. But when you look at nuclear DNA, they're genetically differentiated as both of those are from the pelagic population. So it's really been this suite of methods that's allowed us to sort of tease apart the population structure, the ranges, the spatial use.
Yeah, very interesting, and especially when you have a certain species that is staying relatively in the same area, even if they have a big home range. If you have a tag for maybe 2-3 days, you're not going to get much information. But on these species like the false killer whale and other species that move quite a lot in short periods of time, even a few days, I'd reckon is quite valuable data. Of course, evidenced by the results that your work has produced. Again, do you see, is it a common, or do you do impact studies on the reaction of the animals to the tag? This is a topic that is very interesting, I think, on a global scale, because as tags improve and they get smaller and they get better, I'm always interested in, I think in general, in use of tags, because I believe that if they have a specific impact level that is tolerable, or that is shown not to be very detrimental to a species, then it can provide us very important data. So can you share some light on that, if possible?
Yeah, well, I mean, whether or not it's tagging or biopsy sampling, anytime that you basically cross that skin boundary, you have the potential to introduce infection. And with the tagging studies, we have sort of both short-term and long-term assessments of what the effects are. In terms of short-term, how the animals initially react to tagging, it's very similar to biopsy sampling in the sense that in almost every case you see a startle reaction. So it may be a fast dive, or maybe a tail flick in a fast dive.
Yeah.
In a couple species, we've seen, bottlenose dolphins, for example. We've tagged over 30 bottlenose dolphins, and almost all of them have shown a typical tail flick fast dive reaction. But I think we've had 2 that have had slightly longer term reactions, a lot of tail lobbing or leaping, before they then have settled down. In terms of longer term, we follow up with individuals and are able to document the wound healing. We're able to document reproduction of females if we've tagged a female. We did an analysis that we presented at a workshop in the New Zealand conference 5, 6, 7 years ago, that looked at survival rates of false killer whales and short-fin pilot whales, comparing tagged to untagged individuals in the same group. So both of the species have very strong social structures. So if you see one individual, you're likely to see its entire social group or most of its social group. And so it provided a really powerful way to look at survival rates, because we would look at the survival rates of untagged individuals in a particular social cluster, and then the tagged individuals in that same social cluster, and found no difference between survival rates of tagged and untagged individuals. So we feel pretty confident that the long-term effects are minimal. That said, I think that we have a variety of things in place to try to minimize effects and understand what effects we're having, depending on the species. For smaller animals, we're only tagging them under a very narrow range of circumstances in terms of their behavior and their group size. We have, for the different types of tags, we have shorter darts for small species, longer darts for larger species, in order to make sure that we're minimizing the effects on species that have thinner blubber layers. And I think in any case, any sort of tagging project anywhere where there is a risk to the individuals, people need to make sure that if they're posing that risk on animals, that they're doing it in a case where the results of that research have the potential to benefit the animals. So in our case, with false killer whales, the tagging results have been critical in both recognition of this small resident population. They played a huge role in the designation of critical habitat by NOAA Fisheries, because it was able to identify not just the areas where people were studying them, but the areas that are important to the animals. They've been used to understand the effects of overlap with Navy sonar activities in Hawaii. So it's important that that for folks who are going to start up a tagging study, that there's that mechanism within the regulatory system of whatever country or state people are in, to make sure those results are used to benefit the animals.
That the benefits are outweighing the risks, of course, just not tagging just because. And you mentioned in very well that these tags, they were critical in understanding not only the population structure, but also size and habitat use. And well, false killer whales are, as you said, they are quite endangered in Hawaii, and they are known to interact with fisheries quite often. And I understand also that you use this data, this tag data, to try to establish some measures to protect the false killer whales from fisheries interactions. You set up what is called the false killer whale take reduction team. Could you talk a little bit more about that and the measures that you've undertaken so far?
Yeah. So the false killer whale take reduction team is actually set up by NOAA Fisheries, and it's something. Take reduction teams in the United States are basically a mechanism that can be employed when bycatch of a particular population or species is above a threshold. And so the U.S. federal government is basically mandated that when bycatch is at an unsustainable level, that they should implement a take reduction team to try to find solutions to that bycatch. And take reduction teams are an interesting entity in the sense that they bring together fishermen, scientists, conservationists, and managers to try to work collectively, collaboratively to come up with solutions. So in the case of the Hawaii false killer whale take reduction team, that was started in 2010 in relation to bycatch in the offshore longline fishery. And in Hawaii, there's a lot of fisheries like there are in most areas. The longline fishery is the largest in terms of catch, but it's a relatively small number of boats, 140, 150 boats. And it's primarily an offshore fishery. There were restrictions put in place around the islands in 91 and 92 that basically to minimize overlap between the nearshore fishermen and the longline fishermen. But that team, we met 4 times, 5 times in 2010 and came up with a draft take reduction plan. That plan was we tried to come up with what I would call a gear fix. So changing the gear in a way that would reduce the likelihood of an animal getting injured or killed if it gets hooked on a long line. I'll just see if I have one of the hooks around here somewhere.
You seem to have all the gadget in your office and pull out the tag from the window.
A lot of stuff on my windowsill. That's where I store everything. I don't see a hook at the moment.
It's okay.
The hooks that are used in the fishery are basically, well, 2 types of hooks were used previously. I'm going to just find a hook. I know I have one here somewhere.
Thank you.
I may have to walk further away from my desk. Yeah. So, there were 2 or 3 different types of hooks that were used originally when the team started. A circle hook like this one, but also J hooks. And the thing about a J hook is that when a fish grabs a J hook, it often will ingest the hook. So the hook will end up in the gut of the fish. Whereas a circle hook tends to hook the fish in the corner of the mouth. And false killer whales, when they depredate catch, when they take a fish off a fisherman's line, they would often, but not always, leave the head. So they basically bite the fish right behind the gills and leave the head attached. So one of the things that the team implemented was mandatory circle hooks with the hope or the idea that that was going to reduce the hooking rate. Turns out that didn't work. The hooking rate remained unchanged. Now, the other thing about the hooks that were implemented is that if you look at the wire diameter of a hook, the thicker it is, and also depending on the type of material used, the stronger it is. And there was an idea that, well, the fishermen would say sometimes they would get hooks back that were straightened, that were basically bent. There was enough pressure put on to straighten the hook, and they would just get the hook back, but with no fish on it. So the idea was to come up with weaker hooks that would, if a false killer whale did get hooked, if the hook was weak enough and the fisherman put tension on the line, that it could potentially straighten the hook and allow the whale to get off.
Escape.
So the gear fix that was put in was weak circle hooks. In order for the weak hook idea to work, the rest of the gear, the branch line and the leaders have to be stronger. So the idea was strong terminal gear, weaker hooks. So circle hooks to reduce the number of individuals that get hooked, weak hooks in order to, if an individual gets hooked, to allow it to straighten the hook and get off. As it turns out, our plan hasn't worked. The hooking rate remains the same. The injury and mortality rate remains the same. And so it's a great effort, and people have put in a good faith effort to try to solve the problem, but the plan hasn't worked. So right now, NOAA Fisheries is undertaking a new study testing weaker hooks, with the idea that instead of the hook wire diameter that had been agreed upon or had been implemented by NOAA Fisheries, potentially going for a weaker hook and stronger branch lines to try to go towards that original goal. That said, one of the other changes that was recommended by the take reduction team and was implemented by NOAA Fisheries in 2013 to protect the endangered insular population, because most of the bycatch is with the offshore pelagic population. But one of the changes that went into effect was using our satellite tag data to say, okay, the insular population is using this range around the main Hawaiian Islands. So there was a change in the exclusion zone for longline fishing around the main Hawaiian Islands. There was part of it that would come closer to the islands 4 months of the year, and that was made permanent year round. So that should have reduced interactions with the endangered insular population. So that's one positive thing that came out of the team so far.
Of course, and these are also very complex situations, and will probably need a lot of iteration and testing to see any larger results, let's say. But it's pretty interesting, and I think personally, and I think nationally, we don't have this type of system, and I think it would be advantageous, and it's an example to follow for several countries.
Yeah, I think it's a great mechanism. It doesn't always work, but it's a great mechanism to try to take action towards solving these problems.
Yeah, it's at least a great initiative, and it's also great that you're using cooperating with fishermen in this regard, of course.
Yep.
You mentioned, well, going a little bit back to the whale watching, you mentioned that these projects, they are largely group effort in which many entities and individuals are involved. I understand also that in Hawaii you collaborate with commercial whale watching operators. Could you tell us more about these collaborations and also the importance of these opportunistic platforms for this type of studies?
Yeah, well, the contributions from tour operators, dolphin and whale watching operators are really critical for many of the analyses we do. Our field projects tend to be 2 or 3 weeks at a time off one island or a different island. Because the islands are far enough apart, it's difficult to do work off one island and immediately start work off another island. So we tend to have our projects spread out throughout the year. But over the years, a number of operators have just continued to take photos and contribute photos, and those, particularly off Oahu, I'll give a shout out to the tour operators off the island of Oahu. There's off the west coast of the island, off the Waianae coast, there's really only 6 dolphin and whale watching tours, and 3 of those 6 are sending us photos, sometimes on a weekly basis, if not on a monthly basis. And we just published a paper on trends in abundance of bottlenose dolphins, for example, and most of the photos used in the analysis from Oahu have come from tour operators. So we've done field efforts there off and on over the years, and the tour operators have really allowed us to do those types of analyses. And the same goes for our false killer whale work and our other projects. And on our last field project, for example, we we encountered an entangled Blainville's beaked whale,
Oh,
and we got good photos of it, but we were able to determine that the same individual was seen without the net wrapped around it 14, 15 days earlier by someone that was out on a tour, and a month before that by a different operator, dive company.
yeah.
So those photos really fill in the gaps in a lot of what we're able to learn from our photo ID work.
Definitely, and also something that is, as you said, very important in these oceanic archipelagos, where sometimes islands are super far away from each other, and there's no real way to move. And not only also because certain species, as the false killer whales, they move quite a lot, so they can be in one island and the next day they can be in the other. So having this information from the other from other parts, really important intermediary data, so to
Yeah,
say.
yeah.
So overall, what do you think about working in Hawaii in comparison, for example, in Washington? And do you have any memorable field experiences that you would like to share? Maybe a sighting or anything else?
Yeah. Well, so I live in Washington State, and I get asked all the time why I don't live in Hawaii. Um, and mainly, Hawaii is a great place to work on the water. It's got tremendous diversity, and we're still at the steep end of the learning curve for a lot of species. So scientifically and intellectually, the work there is very satisfying. Um, I still do a little bit of work with killer whales in Washington State, but it's pretty limited. And here there's a lot of researchers who've been working with killer whales in Washington State for a long period of time. So in terms of what sort of contribution I can make as a scientist here is relatively small compared to what is being learned and what has been learned about the populations of killer whales here. Whereas in Hawaii, it's a very different situation with a lot of diversity, and as I mentioned earlier, most of the research had been focused on just one or two species. So, so the contribution that our work makes, I think, is much greater there than it would be here. That said, I really like living in Washington State, despite the weather in the winters. It's going back to being a birder. You know, in our backyard, we have 20 different species of birds, 19 of which are native on a daily basis. Whereas in Hawaii, it's it's isolated archipelago where introduced species have played on land, have played a really dramatic and detrimental role on the native wildlife and the native bird populations. So it's not actually a place where I want to live. Which surprises a lot of people. That said, I really enjoy working there, and just got back a couple weeks ago from a trip last year with the pandemic. We had one trip that I made just before things closed down, and then I managed to get back in December for a field project off Lanai. The work is a lot of it is. Well, Hawaii is very low density. It's an oligotrophic system, which means in any one day we may not see very much. A couple sightings a day of all species is typical. Occasionally we have days where we see nothing, and other days where we have 6 or 7 sightings. Our field project often comes down. Our field projects, you know, how much we accomplish. Sometimes it's just slow incremental increases in the number of photos we get of a particular species. And then other days we have amazing encounters where we're able to witness some really unusual predation, false killer whale predation on mahi-mahi, where they're sharing the prey and throwing the prey up in the air, or trying to get fish away from floating debris, or cases where we're able to collect a bunch of biopsy samples from known individuals in our catalog, and none of them have ever been biopsied before. So we're sort of building on that long-term assessment of relatedness and mating systems and so on. So, yeah.
Very interesting. And yeah, I imagine that this is kind of, some of the surveys are quite a bit of a lottery where you might not see anything, or you might just stumble upon something that you've never seen before, and you will learn things that were previously unimaginable in some cases. Now, in more of a personal questions, if that's okay with you,
Sure.
as we saw previously, you have a son. He was born a few years ago. I don't actually remember what year he was born in, Bryson.
2016.
2016. Okay, so he's about 5 years old right now.
That's right.
Of course, having a son is something. Having children is something that changes. Would change anyone. So, would you say that Bryson has had an influence on the way you approach your work, or in? Yeah.
Yeah, well, it makes it much harder to go into the field. We've been lucky in that I've been able to convince my wife to bring Bryson to Hawaii. He's been there 5 times, I think, now, which is a bit crazy. But having them come for field projects has certainly made it a lot easier, and more desire to spend more time at home. But you know, it's interesting, the perspective of trying to make sure that the work is having some long-term meaning, some long-term impact. You know, I've always said when talking with fishermen that, well, you know, if I had kids, I'd want them to be able to go fishing when they're 20 years old, and for there still to be fish left for them to catch. And a lot of it is, you know, trying to make connections with people, trying to make connections with fishermen, trying to show that there's a common interest in conserving and protecting fish populations and whale populations, both for the whales and for the people in the future. So it certainly has changed my thoughts on that over time.
Yeah, and that probably made it more personal, because you're looking forward for Bryson to experience a lot of the things that we were able to experience. And what about when you were younger? Who were your idols when you were growing up? You said Mike Big was your mentor. Who inspired you? Who was kind of your role model, or if you can call it that?
Yeah, well, you know, when I was a kid, my mom used to take me to every zoo and aquarium everywhere we were, either in Victoria or whenever we took trips anywhere, and I grew up watching nature programs as they were back then. Mutual of Omaha, Wild Kingdom, Marlon Perkins. You know, it's certainly having a childhood where I could just go and tide pool and look around and see crabs and various things so easily influenced my long-term interest. And you know, our backyard here, we do all sorts of things to benefit local wildlife, benefit snakes and amphibians and birds, and building structures, bird boxes and structures for wildlife, so that we can take Bryson. Yesterday I caught a snake in the backyard and showed it to Bryson, and you know, trying to teach a 4.5-year-old about different birds and different plants, and just you know, instilling that interest in nature. He definitely is fearless when it comes to things like spiders and bugs of all sorts. So
Of course, and he's being exposed to that very early, so more likely to be interested and surprised rather than anything else.
yeah.
And what about any plans? Do you have any specific plans for the future? Of course, these studies and these projects, they're very long-term, and well, they never end. Actually, we never reach a point where we just say, well, we know everything. So do you have any specific future plans regarding research, maybe new areas of study, new methodologies?
Well, I mean, one of the things that came out of the pandemic last year is we adopted a slightly different strategy than our typical um, uh, taking a team to a particular island for a 3-week period. A couple of our main project crew are based in Hawaii, and one of them a couple of years ago purchased a dolphin and whale watching company. So last year, when all the business shut down, he was able to get out in the field, I think 26 days over the last summer, and continue to deploy tags, collect biopsy samples, do photo ID, and sort of continue the work in a way that was more taking advantage of the good weather opportunities. So even though we just did an intensive field project, Colin and his company, Captain Zodiac in Kona, they went out on Sunday to do a survey looking for false killer whales. They didn't find any, but putting the time in. And so that work is going to continue over the next year when there's the flexibility to get out when the weather's particularly good. We have a couple field projects planned for the rest of the year, one in August off of Kauai, and then another one in the fall. And in the meanwhile, there's just a lot of people in the office that are, or their home offices that are working on photo ID catalogs of various species, data analyses. We have a lot of data sets, and we could stop doing the field work today and spend just the next few years working on analyses and writing, and so a lot of it is trying to figure out how to find the time to continue to make progress on those. So.
Definitely, yeah. Being in the field is only a small part, a very small part of the work.
Yeah.
Yeah, so I think that was a really great deal of insight from you, from your career and also your personal experiences. I also would like to, for anybody watching, to let you know that you can look more into all of this information and all of the publications that came from these studies at Cascadia Research Collective. You can find it online pretty easily. It also has a very nice section for those of you who are looking into starting a career in the marine field. Dr. Robin has provided quite a nice list of steps and advice for these little steps just to get into this field, what you should do, and some tips from, of course, a professional. Dr. Robin, I want to thank you very much for taking your time for this interview today. It was delightful for me, and will probably be for everyone who watches this interview. As you, of course, you're a person with a lot of experience, also an inspiration for a lot of us, and also a role model. I don't know if you want to say any last words or leave any information available. Please do so.
Yeah, no, well, thank you very much. One thing we touched on at the beginning was, if being a birder, it's a really, your point of birders make good observers is a really important one. I think that it's just an example as well that people who are just getting into the field or studying something should try not to be too narrowly focused. Pay attention to the other things that are going on around, whether or not it's birds or fish populations, oceanography. It's the broader approach, I think, is really important, and people need to not be too narrowly focused in what they do. And it may not seem like a good short-term strategy if you're a graduate student, but in the long run, it's critically important.
Of course, and the environment is all connected, so connections are there even if we don't know them, and we're just benefited. Our analysis and our look of things is just benefited the more we know. So definitely great advice. And so with this, we're going to finish our interview. Again, thank you very much, Dr. Robin, and good luck with all of your future projects. I hope you have a lot of success, find a lot of rare species, deploy a lot of tags, and publish a lot of important data.
Well, thank you.
Thank you very much.
Good to talk.
Good to talk to you.
Okay, take care.